Hello, I'm Luca Comai, a professor at UC Davis, and by studying this video, you're going to learn and understand four important principles of DNA structure: 5' and 3' polarity in DNA, bases and their pairing, Chargaff rule, and DNA melting. Let's start with polarity. Considering single-stranded DNA, one end is the 5' end, the other is the 3' end. The meaning of this nomenclature is shown using adenosine monophosphate. In this 3D model, the oxygen atom is red, hydrogen is white, phosphate is orange, and carbon is gray, and nitrogen is blue. The largest group is adenine, the nitrogenous base. The next largest group is sugar deoxyribose, to which is attached the phosphate. Because deoxyribose is the next to the largest group, its C locators are given the prime designation. The C closest to adenine is 1', the next is 2', and so on until the 5' carbon, which is connected to the phosphate through an oxygen. Important, the 2' carbon carries an hydrogen group instead of an hydroxyl. Therefore, this is deoxyribose. A nucleotide polymerizes, that is, forms covalent bonds with other nucleotides through two regions, the 3' hydroxyl and the 5' phosphate. Consider a single DNA strand. It's formed by polymerization of nucleotides. You can think of each nucleotide as a Lego piece with two snap buttons, one 5', the other 3'. We will learn that all organisms polymerize DNA in the same way. They snap a new nucleotide to the 3' of the other. A single DNA strand can pair with a complementary strand through hydrogen bonds that are formed between bases. Complementary strands pair oriented in the opposite way. In summary, double-stranded DNA is formed by two paired single-stranded DNA molecules. Their 5' to 3' orientation is opposite. The two strands are said to be in an anti-parallel orientation. Let's consider the concept of base pairing, purines and pyrimidines. In each base pair, one base is a purine. Here are the two purines in this DNA molecule, guanine and adenine. You can tell they are purines because of their double ring structure. The other base is a pyrimidine. Here are the two DNA pyrimidines, cytosine and thymine, identifiable by their single ring structure. C and G pair through formation of 3 hydrogen bonds. T and A pair through 2 hydrogen bonds. This is because this hydrogen in adenine is not suitable for hydrogen bond formation. An adenine can only form 2 hydrogen bonds. What is Chargaff rule? It states that in double-stranded DNA, the fraction of C is equal to the fraction of G, and the fraction of A is equal to that of T. How did Erwin Chargaff come up with the rule? He observed it by studying the composition of multiple genomes, shown in this table. He isolated DNA from this species, hydrolyzed it, and measured the amount of each base in the samples. For example, in wheat, G and C are both about 22%. A and T are about 27%. In grasshopper, G and C are about 20%, A and T about 29%. If you study this table, you can see another discovery by Chargaff. The DNA of different species has different CG to AT ratios. That is, different CG content. When first discovered, Chargaff rules seemed mysterious. Watson and Crick were able to interpret it. It meant that in DNA, in the double helix, C paired with G, and that A paired with T. It is common that exam questions require you to solve DNA composition using Chargaff rule. Let's try a couple of examples. Here's the first problem. A double-stranded DNA W contains 30% A. What percent of base are C? We know that the percent of A is equal to the percent of T, which is equal to 30. Thus, A plus T is equal to 60%. It followed that C and G is equal to 40% and C is equal to 20%. The answer is 20%. You can also visualize this by using a 10 base pair double-stranded DNA that has 6 A's, 6 T's, 4 C's, and 4 G's. As long as the number is 6, you can put the A's on either strand. The same is for the C's. Both of these double-stranded DNAs have the same composition: 6 A's, 6 T's, 4 C's, 4 G's. Out of 20 total nucleotides, this is 20% C. So you get the same answer. Let's try another problem. One strand of double-stranded DNA R has 20% A. The other one has 70% A. What percent of double-stranded DNA R is made of C's? One can solve this problem in at least two ways. First, you calculate the total AT content at the double-stranded DNA level. The percent of A is equal 90%. Dividing by 2 to account for the double-stranded structure, the percent of A is equal to 20 divided by 2 plus 70 divided by 2 equal 45. Then A plus T is equal to 90. The percent of C plus G is equal to 10. And the percent of C is 10 divided by 2, or equal to 5. The second method uses our 10 base pair long DNA with 2 strands. Here we're going to call them respectively Watson and Crick. Watson has 2 A's. The complementary Crick region has 2 T's. Then let's add 7 A's to Crick and the corresponding T's to Watson. We're nearly done. The last base pair must be CG. We can then count 1 C out of 20 nucleotides, or 5%. The last concept we're learning is that of DNA melting, also called denaturation. To understand what it means, let's draw a double-stranded DNA molecule, starting with the blue strand. The red complementary strand has a red purine corresponding to each blue pyrimidine, and vice versa. There are 3 hydrogen bonds between C and G, and 2 between A and T. Therefore, you can identify the base in each base pair based on the ring number and the hydrogen bond number. We now heat the solution containing this DNA to 95 centigrades, close to the boiling point of water. The double-stranded DNA melts, that is, the 2 strands separate because the heat energy has broken the hydrogen bonds. Because CG pairs have 3 hydrogen bonds, they are harder to break than AT pairs, which have 2 hydrogen bonds. Melting DNA with a high CG content therefore requires higher temperatures. Although at 95, they all typically melt. If we cool the DNA solution to 55 centigrades, the 2 strands can form hydrogen bonds again, and the DNA undergoes renaturation. That is, the 2 strands, if they are complementary, zip up and form a double-stranded structure. This process is also called DNA hybridization, and the complementary strands come together perfectly because they base pair perfectly and form double-stranded DNA. In conclusion, you should now understand 5' to 3' polarity. You should recognize the bases and their pairing mode. You should be able to work with Chargaff rule, and you should understand DNA melting and the process of DNA hybridization. Yes, DNA is a complex molecule, but if you memorize its structure, its properties will make sense, and it will be easy to recall them. Importantly, these properties are the necessary foundation to understand the molecular biology of DNA and its manipulation.